afternoon.
Morning.
My guest, Mr. Adam Quicquaro, Pharm.D. How are you doing today?
I'm doing great. I'm happy to be here. Finally part of Career Day.
Yeah, I'm really excited to talk about your career. We might have to do a second part because we only have about 50 minutes right now, but that sometimes time constraints are good, as you know. So can you introduce what's your title? What's your job title right now? And if you choose, you can talk about the company you work at or not. It's up to you.
Sure. Yeah, sure thing. So my current title is Associate Director, Clinical Development Scientist. I'm a pharmacist by training, as you said, Pharm.D, but I decided to take that in a slightly alternative direction and focus more on development as opposed to clinical practice.
Development of?
The company I work for, sorry, what?
What development of what?
Medicine, therapeutic products.
Okay.
Which I'll talk about a little later because I'm working on both a therapeutic product and also a bioassay, which is really cool. Nice. But I'm working at Alexion Pharmaceuticals, recently acquired by AstraZeneca. They are a company that focuses,
AstraZeneneca of the vaccine fame.
Yeah, they're double-stranded DNA vaccine. Yep. But AstraZeneca is more of a like an ambulatory care, a chronic care, a mainstay condition or disease company, whereas Alexion Pharmaceuticals, they're a rare disease company, which is really cool because I find that working in rare diseases is really altruistic. Someone who has a rare disease is, out of 200,000 people in the United States, probably one of the only ones. That's like the prevalence rate for a rare disease. Every country and
1 in 200,000 is it?
Yeah. Every country and disease and region of the world has a different definition. I'm sure Dr. Chardins could address this much more eloquently than I can, but different parts of the world define a rare disease in different ways. The U.S. says it's around 1 in 200,000.
Yeah, that's pretty rare. So let's backtrack a little bit. How did you decide to go into pharmacy? How did you pick that career? I have a memory of this. I want to see if it matches up with your memory. My memory is we took like a kind of career aptitude test in high school, like sophomore year, maybe. And I remember you got your results back, and everybody was looking at the like starting salary for different jobs, and pharmacist was one of the highest starting salaries on the list with like 100K, I think. I don't know if that was when you started thinking about pharmacy or not.
Oh, well, it wasn't the inception, but it definitely was. I'd be lying if I said it wasn't appealing. Definitely the compensation's appealing for sure, but I think that my interest in pharmacy, my ties in pharmacy, personally, I think goes really far back to kind of like two different paths, I think. And I know you know my father, you know my dad. Most people probably listening to this might not, but he's a software engineer, and in our basement he has a lab, and that's where he does all of his whether it be hardware, software, you name it. That's where he does all of his fun little experiments and works on his own products, family products, tries to fix them, make them better, hack them, do whatever. When I was a kid, I thought the lab was the coolest thing in the world, and I was so obsessed with being like an inventor and being just someone who was a designer, a development person. And that was really honestly the back of my mind, though. I never really tied that into my life until recently with my current career. But to your point, which I think you might be alluding to, when we were in high school, I was so fascinated by definitely biology and neuroscience as a whole, but more so just the application of medicine and just the art of pharmacy. The tying in of chemistry, biology, like so many different disciplines. To me, how something so small could have either a detrimental or beneficial impact on the body and a person's condition or quality of life.
So how would you
tie it all in together, like my subliminal desire to be some sort of inventor or scientist, and then my passion and love for medicine and pharmacy just tied in really nicely together, which led me to my current role in R&D and clinical development for a pharmaceutical company, biotech company.
Cool. So what are some of the experiments or inventions that you remember your dad doing when you were a kid that stick in your mind?
So we had an Xbox growing up, but it wasn't a very typical run-of-the-mill Xbox. He was part of a very small group of software engineers around the world who kind of came together and said, there's a lot of Linux-based underwritings, I believe, in the code, and they thought that they could make something really cool, something very, I believe the term is called, not open concept, but something along the lines of like being able to just
source?
Open source, just being accessible by the average typical daily user. And so they kind of hacked the original Xbox and were able to
they jailbroke it.
And then you can get any mod, skin, like game if you wanted, if it was available out there. It was that was one of the first things I remember working on, which was so cool as like a fourth grader.
I remember people used to talk about jailbreaking all sorts of things. Okay, and another question: if you had to give like a definition of pharmacy, what would it be?
Oh, that's a great question. My definition would definitely vary from your typical pharmacist, but I'd say that the practice of pharmacy is the application of drug management, drug utilization management, drug utilization review, just basically that last baton pass from the physician to the next healthcare professional in the patient's journey in treating their condition. It's once you're diagnosed, yes, the physicians are incredibly well knowledgeable in regards to the treatment guidelines and what you should be on and what happens next. But when you start getting into those minute questions, those minute details about, is this going to interact with everything else I'm taking? Is the side effect I'm experiencing common? Is it really unique? Those are some things that kind of fall in line more so with pharmacy. And also deciding, like, I know you and I were talking a little bit before this interview, the generic versus brand. A lot of physicians don't really know the distinction between those.
Right. Interesting. Do you want to talk a little bit about like the history of pharmacy? Because I find this kind of fascinating, what I've come across, the history of like the discipline, the science of drugs.
It's cool because like around the world, there's just different historical timelines for pharmacy.
Right.
If you go to like the Middle East and in Africa and Egypt, you'll see that there's the I think it's called the Eye of Horus or something. It's kind of like the foundation for the RX symbol, which in Latin means recipe.
Yeah. Oh, I didn't know that.
I think it's the Eye of Horus, but it's found in Egypt and not only in Egypt, but a lot of cultures around the world, like revolutionary times in the United States, England and Europe,
like occult
even
stuff,
Russia.
right?
The pharmacist and the physician were like the same role. You had the
alchemist, right?
Yeah, exactly. Yeah. I mean, it's really cool, just the timeline of medicine and how it's progressed.
Would you be able to say when pharmacy went from this kind of like occult art to more of a scientific discipline? Or is that more of a historian?
I think it was, I mean, I could be wrong, but in my opinion, I think it was around that early time frame of the 20th century when we started pushing into regulations and the FDA and the USP, United States PIA, and the Curies of the world, where you're really concretely making the chemistry table more finite, and we're starting to tie in more of these effects. This is all what we refer to as small molecules, tablets and solutions and injections. It has nothing. There wasn't many biologics back then like we have now.
No.
I'd say it's around that time where we see modern medicine and modern pharmacy kind of coming to fruition.
Yeah, I was just reading the other day that around the turn of the century is when like Louis Pasteur came around and like definitively proved like the germ theory, that a lot of diseases are caused by microorganisms, and you can pasture like superheat stuff or treat it with disinfectants to actually kill germs, and that's when you start to see the human population like shoot up, right? That's one of two reasons why the people started living longer, and very cool. So
there's so many gods in pharmacy, and a lot of people. Louis Pasteur, like for example, if you're a pharmacist who works in infectious diseases or compounding, Louis Pasteur is your hero.
Yeah. So you went to college for pharmacy. Can you talk a little bit about your experience at university or college, whatever you want to call it?
Yeah, sure. I'm one of the few people who, the technical name of my school is MCPHS University, which stands for Massachusetts College of Pharmacy and Health Sciences. I just like to tell people I went to Massachusetts College of Pharmacy because it's its own little thing in the grand scope of the university. It's in Boston. It's not too far from where you and I ran and participated with New England's and saw those championships and all that.
The Reggie Miller Center?
Yeah, it's not too far from that at all. So it's a cool little area. Really nice part of the city, really quiet, not too hustle and bustle like some areas.
Really?
But it's a unique program. I went through 6 years of school. I did not get an undergraduate degree. I went through the accelerated program. I'd say the majority of pharmacists have an undergraduate degree. So after 4 years, they then do 4 years of pharmacy school in the doctorate program. I had a very blended model. For better or for worse, I don't have it. Like I have a cousin actually who is part of another pharmacy program in New England that will give you your undergraduate degree. You get your bachelor's in pharmaceutical science after completing a certain amount of credits in a certain time frame. I didn't have that, for better or for worse.
Yeah, your school, that's more like the European and South America. Well, most countries in the world follow that model more, where if you're going for like a medical doctor or any sort of like advanced degree like that, you don't go for a bachelor's first. You just kind of enroll directly, and it takes less time. Whereas in the United States, you have to take all these like gen ed requirements and get a bachelor's first. So like it's a very American way of doing things. But anyway,
it's interesting you say that because I had I'd say definitely around 50% of my class was foreign.
Yeah.
International student.
So what was it like, if you can remember, going into college in the fall of 2012 and starting a pharmacy program? What was your experience in the beginning? And then how did that change, like, as you progressed through the program?
Yeah, I think that Holy Cross really gave a unique perspective on education and how to approach it. It benefited me in many ways, but I think there are some things that I definitely was missing. I found a lot of courses challenging, even though they were basic, run-of-the-mill undergraduate courses. I found like chemistry was challenging to me when I had to actually go into the labs.
Mr. Kaza was not the best chemistry teacher, to be honest.
But hey, he was an inspiration for my career choice, without a doubt.
Did you find that the professors were the type that didn't really like feel like they had to feed you spoon feed you? They were more the type of professors where it's just like, I'll give you the information and it's your job to learn it on your own kind of thing?
I had both in college.
Yeah.
I really did. I had some who were incredibly dedicated to the development of the student and making sure you understood the material at its heart and its essence, because that's kind of like what I do now for my job. And then there were others who were just like, this is the guidelines, memorize it. I hope you never see a patient like this. Good luck.
Oh, goodness. So what was the curriculum kind of like? What did you start off taking?
The curriculum was very run-of-the-mill in the beginning. You had your chemistry, your biology, your calculus and whatnot. But then when I hit my third year, organic chemistry second year, it followed the path of the undergraduate process. But when I hit my third year, it started to really diverge, where I still continued on that advanced biochemical route. I took biochemistry 1 and 2, but I was also in my third year started to take pharmaceutics. I was starting to take classes that were more related to my practice.
So pharmaceutics.
And then it continued. So like in my fourth year out of the 6 years, I started taking really advanced courses like medicinal chemistry, pharmacology, what have you.
So, keeping in mind our audience is not pharmacists, what are some little things that you would learn in those pharmacist-specific classes as opposed to like general chemistry? So what are some things you learned in like pharmaceutics or pharmaceutical science, medicine?
This right here is beautiful coffee. Whereas Cassandra's family would call it espresso con panna because it's cream, milk, and espresso. So just taking something simple like that and going down to like the microscopic level taking the foundation of organic chemistry in chemistry, so inorganic, organic, taking those principles and thinking about what's inside of there, how it's going to go through your body,
right?
What's going to happen to it once it's in your body, what is it going to do to your body, and when's it going to leave, or is it going to stay there? How long is it going to stay there? Just
so many like what's the half-life, or what do they call it? How long the drug stays in your body?
Yeah, that's one term. Half-life is an awesome parameter in pharmacokinetics. Awesome parameter that can, with that one parameter, most drugs with that one parameter, you can tell a great story about what's going to happen to the patient, how they're going to feel. Yeah.
Isn't it caffeine? It mimics what's that? The nucleic acid, the
adenosine.
Yeah, adenosine. Doesn't caffeine mimic that? And that's like all we really know about it, right? Because it's like we don't really know much else.
I mean, my basic recollection, like my go-to, like that's the thing is pharmacy school, or just being a pharmacist in general, you have to have like a top line understanding of a lot of things, a lot of different chemicals, and then you also have to have a deeper level for those more intricate situations. But caffeine, like we know that it'll displace certain compounds on adenosine receptors, and that's what causes it to give you upregulation, which is why you have to keep drinking more every week. You get the same feeling.
Do you think it can be used to make ATP?
Yeah, wait, what?
I was just thinking, do you think it can be used to make ATP, adenosine triphosphate, the energy molecule?
I don't know.
That'd be wild.
That would be wild. I do know that it causes, so like, this is a great example actually of like, you're asking like, what did I learn in school? Caffeine, right? That could bind, I could think of 3 different areas in the body. One, it's going to give you an increase in blood pressure because it's going to constrict your blood vessels. Another region, it's going to increase your heart rate. And then another one's going to help you with your cognitive function.
Interesting. What are some fun facts about drugs? Any drug that you can think of? What's like one of your favorite fun facts about drugs?
I remember when we were in high school and I was working at Woodbury Drug Company, the pharmacist there, really interesting tidbit that said, everything in the world is a medicine, but the dose determines its like toxicology. It determines its toxicity, or like how bad it is for you. Everything in the world, this is her saying, not mine, but it's an interesting tidbit. Everything in the world has some sort of therapeutic benefit, if you will. It just depends on how much will hurt you.
Yeah, even opiates, right? I used to sometimes say to like my middle school students when I was a sub, I'd say, everything in moderation except for opiates. But then I realized even opiates, if you're truly using opiates in moderation, then it's okay if you're using it for like pain management under the guidance of a doctor. Obviously, shooting up fentanyl is not moderation, right?
Yeah. Actually, just learned this the other day. So one of my colleagues is a neurologist in clinical development. And they used to, when they were a resident, they were in surgery and in the operating room. And I said, what's the relationship with opioids in the brain when you have a patient under anesthesia? When you're under anesthesia and your consciousness is gone, you're not going to react to pain. You're unconscious. And I said, does the brain have some sort of signal that responds to pain under anesthesia and unconsciousness? And the neurologist said, definitely. I'm like, so that's why you give opioids to someone who's unconscious, so their brain is more comfortable while you're operating on? He's like, definitely. I'm like, cool.
Yeah, that's awesome. I think otherwise you could potentially wake up, right? Like if you're sleeping and
if the pain's bad, I guess,
if you're sleeping and I shocked you, you would probably wake up, So I know it's like a cocktail they give for anesthesia, but one of them, also opioids kind of make you fall asleep, also make you sleepy. But what is one of the most challenging and one of the most rewarding experiences you remember from college? So it could be a class or like a particular thing that you did.
Yeah, okay, so this is really cool. There's a class called Pharmacoepidemiology methods, which I don't know why I keep going back to Dr. Desjardins, but he could probably talk about
we love Mike Desjardins on this podcast.
We do. I passed that course by the skin of my freaking nose. Like, I just barely passed it. And honestly, it's probably the most most important class for what I do right now.
Oh.
That particular course talks about your sample size, how to detect statistical significance, how
like a research
covariance, confidence intervals, all that crap. But it's funny because like I have a great team. I have a biostats team. I have pharmacovigilance team. I have all these people that help me with my weaknesses. And my job is to really kind of support the clinical goals and try to show that the intervention is performing how we hypothesize.
That's funny because a lot of that stuff overlaps with like psychology research methods. So like that was more my area of study, and you would learn all those terminologies in a psych research methods class as well. Thing is, as a psychologist though, a lot of times you're expected to like do your own stats, program your own. You have to be like a jack of all trades in a sense.
Oh wow.
Okay, so let's talk about your work now. So you're working for a rare drug company called Alexion, right? And what do you do? What's your day-to-day like?
My day-to-day is kind of like why I love my job. When I was, if you don't mind, I'm going back a little bit. When I was on my clinical rounds, I definitely enjoyed being at the bedside of the patient, helping the physician tackle why this patient's taking the meds they're supposed to. They're following the guidelines, but something's happening. Why? So we look at their, not only the medical profile, but their treatment regimen as well, and try to come to some answer. I loved all that, but it became pretty like routine after a while. And I was always asking myself when looking at particular therapeutic products, how do they get here? Like, how could I be a part of this? And that's how I found the pharmaceutical industry and fell in love with it. Because remember, my early ties as a child to want to be an inventor and be some sort of scientist. This was a really nice way to marry my two passions. And so my day-to-day changes because I'm overseeing different studies. In some studies I'm not even overseeing, some I'm just supporting, some I'm just there, like almost like on call. Like I own a small part of that study, and if a regulator or a physician that's part of the study needs answers or needs to discuss it or talk through something, like I have to be there. But then again, I'm also overseeing certain studies, so I could be interacting with the physician, I could be interacting with my team one day, meetings here, meetings there. Some days I don't want to talk to anybody. I want to deep dive into literature, strengthen my hypotheses, or find more information to support why we're seeing some weird stuff in the study that we didn't anticipate.
So you get to read literature as part of your job. That's cool. That's like a true researcher kind of job.
I'd be so lost without it.
Yeah. So it's almost like you're working in academia. You're doing a lot of research.
I love that comment, because I work with so many people who were titans in academia, and they reached a point where their love for whatever they were working on also met some sort of limitation in grants or funding or whatever. And as you're probably very well aware, the pharmaceutical industry has a lot of cash flow to go around.
Oh yes.
And so these titans kind of come to the industry, and they're known. They're called KOLs, key opinion leaders. Okay. So they're known for what they do and what they are investigating. They come to the company, and then they get this huge influx of cash to do whatever they want. And for better or for worse, either supports their findings, or they have to keep going down a different rabbit hole.
Right. So they have like kind of more freedom. I think academia can be limiting, like you said, because of money, and also I don't know, the university might just not support if you want to switch from studying one thing to another thing. So you end up getting like pigeonholed, studying the same specific thing for your whole career, which kind of sucks.
When I go back to school and I give talks in some of the classes, and they ask what's the biggest difference between research and academia and industry, my personal take on that, people are free to form their own opinions, but my personal take is that in academia, you can be, both types of research are definitely contributing to the greater good of science and knowledge and pursuing and forging forward. But I think that with academia, because of some of the limitations that you just discussed, I find that the nature of a lot of the research might not be focused on intervention. It just might be more so about like discovery or just moving forward the idea of what we know.
What they call pure research as opposed to applied research, right? So your research is much more
like
applied.
That's not a registration trial, not testing something super concrete. Yeah.
So applied research is usually Yeah, applied research is usually trying to find like a cure for a specific disease. Like you have a specific problem and you want to solve it. Whereas pure research is often just like, oh, I wonder, like for instance, I wonder how caffeine works. And it doesn't seem like a dire need, right? There's not too many people who are like caffeine is playing a life or death role in their life. But the cool thing about pure research, and like my research methods professor used to talk about this in psychology, is that a lot of important discoveries are made through pure research that then have applications down the road that we did not foresee. So like if you do investigate just something kind of out of curiosity, sometimes you'll come across like principles of pharmacology that can save lives later on. So both are definitely important. I think that's definitely true.
I love that. I think that's you articulated that way better than I could, because the product I'm working on right now, the indication we're pursuing is for Wilson's disease. But the current product is also being looked at by Mount Sinai and other academic clinicians for breast cancer, for ALS. It's being looked at for other other conditions where there is proof of concept because of the reasons you just said.
So yeah, this is all like I do want to talk to you about a couple specific things if we have time today or in another episode, and those would be just a preview. Some news about Humira and the company Abbvie, and also the vaccines that have been put out for COVID and how they actually work on a molecular level. Because I remember you telling me about these last summer before there were vaccines, telling me like we might see an RNA vaccine for the first time in history. So that would be really cool to talk about. But before that, I want to talk more about your day-to-day thing. So can you talk about, are you allowed to talk about one of the studies that you're working on? And like, yeah.
I think at a high level I can. There's some things that I have like patents pending and publications pending, so I can't.
Yeah.
But at a high level, this is all accessible. If you go onto Lexic's website, you can see that their product 1840 is being tested for Wilson's disease, and we're currently in a phase 3 trial.
Okay. So what is Wilson's disease?
Yeah, Wilson's disease is a nasty disease, but it's one of the most common rare diseases out of all the rare diseases. What you'll typically find is the patient has either liver hepatic condition, like really bad cirrhosis, or a NASH type form.
Fatty liver?
Or, sorry?
Do they get like non-alcoholic fatty liver?
They're often diagnosed as having that. But then when you start to see that they have neurological or psychiatric complications mixed with that, that's usually when the clinician says, okay, this is probably Wilson's disease. When the neuropsychiatric symptoms are separated from the hepatic symptoms, the liver condition, that's when you kind of lose these patients. It's not until you take a blood draw and you say, oh my God, their copper levels are paradoxically super low, when it's an actual condition of copper toxicity where the tissues are just overwhelmed. It's just the copper's been deposited everywhere, and it really affects your metabolism, but most prominently it affects your liver and neurological status.
So is that something that doctors usually would test for, copper levels in your blood? Or is that like the doctor has to kind of suspect Wilson's disease to order that test?
It depends on your region around the world. So in the United States, we hate drawing blood on children. We do not draw blood on children until there's a problem. In Japan, they'll draw blood on, I think, 5 or 6 year olds routinely as part of like not a state program, but just kind of some sort of procedure,
like a standard, or whatever.
Yeah, and they catch Wilson's disease better than anyone else in the world. They catch Wilson's disease like insane. And you could talk to almost any resident, they're gonna have one question on their boards about Wilson's disease because it's one of the most common rare diseases of all of them.
What's the age of onset?
So the age of onset is adolescence, but we're finding that some are escaping that and becoming more older as they get diagnosed. But mostly adolescents.
And what are some of the psychiatric symptoms that are commonly seen?
You can get psychiatric, that's a good one. I'm more familiar with the neurological complications like the wing beat tremors, the dystonias, the dyskinesias, like the physical. But the positive symptoms of the psychiatric condition, I'm not as familiar with. I think it's just mostly that.
So they get like tremors and dystonia, meaning like they don't have good muscle. So that's like the stuff that your doctor, when they test your reflexes and are like, push against me, that's what they're testing for, right?
Yeah,
Gotcha.
but I guess this is more neurological and psychiatric, but their speech goes quickly. Their speech will go really fast. They start as like a waning, and then all of a sudden a slurring, and all of a sudden just like, it's really bad. Yeah, their speech is one of the first. It's not so much psychiatric, but more just a neurological complication.
Gotcha. Although psychiatry and neurology could be considered the same discipline, right? Because the mind and the brain are the same thing. But in my opinion,
I like to think so, but when I talk to certain neurologists, psychologists, they all have their own opinions. So I kind of keep away from
that's that old mind-body dualism thinking that the mind is separate from the brain. But modern neuroscientists, we say as a statement of belief that the mind is the process of the brain, and mind is what the brain does. So in any case, what's going on at the microscope at the cellular level with this copper toxicity? Why do they get copper toxicity and have low levels of copper?
Sure. So you mentioned ATP earlier.
Yeah, adenosine triphosphate.
Yeah, you mentioned ATP. So ATP has I'm not a geneticist, but a very wide array of expression. There's a lot of different I don't want to say phenotypes, but it's a protein that can be expressed in different isoforms and have different functions. ATP7B deals with your transferring of copper out of the cell onto proteins that safely carry it in the blood. ATPA deals with its influx, not its efflux.
So it's a transport. It can be a transporter for copper, is what you're saying.
Yeah. In this particular situation, yes. And it can have a mutation, whether it be there's so many different kinds. There's autosomal recessive. There's compound heterozygous. We're scratching the surface with genomics. Let me tell you.
Yeah, sure.
There's literally by the year, our understanding of Wilson's disease and many other conditions just changes with genomics and genetics. But so if you have a mutation in ATP7B and you can't properly or safely load copper onto transport proteins in the blood, it just has to be spilled over on albumin and then transferred into your tissues around the body, unfortunately. And if you think about copper, it's a metal. It's
heavy metal.
There's a lot of
It's a heavy metal.
It is. Yeah. And if you think about, we can go back to the principles of chemistry and just the valence and how electrons will interact with the tissues in your body, if it's not safely carried around, all that oxidative stress everywhere, it's going to wreak havoc.
Yeah. So so you're basically saying because the copper is not transported into the blood and carried away in the blood, it stays in the tissues and does damage to the tissues, right? So that's why it gets moved around in the blood, but very sloppily on albumin. Albumin is a godsend in of itself because it picks up so many things that aren't supposed to be there, but it doesn't do it well. It's kind of like a tugboat. It's just whatchamacallit? But you said they usually have low copper levels in their blood tests, right? Paradoxically, yes. So I don't want to go into the weeds too much, but that protein I was referring to that's supposed to be loaded onto is called ceruloplasmin. And if you don't have copper loaded onto it, it degrades. So ceruloplasmin is like 80% of total copper stores in your blood. There are other proteins that carry copper, but ceruloplasmin is the big dog. And if you don't have copper going onto it, it's degrading. Your total copper is going to be very decreased relative to you or me. You know who's really good at storing copper in their blood? Who? What? Crustaceans, lobsters, crabs. Oh, cool. Yeah. So that's awesome. You didn't know this? They don't use hemoglobin to bind oxygen. They use a copper compound. So they have blue blood as opposed to red. It's like they don't use. Serum? Is that what it is? I don't know what it is, but they don't use iron to bind oxygen. They use copper to bind oxygen. So their blood is blue as opposed to red. So this is why you should have biologists on your team, because I wonder if you could derive something from lobsters to help cure. Well, I know this is what I do know. I can speak to this. So there are certain. This is where the clinician part of me comes in. So like, if I had a patient with Wilson's disease, I would advise them to stay away from shellfish and crustaceans and whatnot, because that could alter their copper levels. I know that. I just didn't really think about.
blood content, but
yeah. Okay, so we talked quite a bit about Wilson's disease. So what are you doing with the clinical trials? You're coordinating clinical trials of an assay, is that right?
Yeah, so I have two things. So I'm supporting the phase 3 trial, the pivotal trial that will hopefully get the therapeutic product approved by the regulators and demonstrate that.
So phase 3 is humans, is that right?
Yeah, phase 1, 2, and 3 are all humans. It's just different objectives as you go up the ladder, and you change the risk threshold too, if you will. But you change the risk threshold because you're proving that it's safe and effective as you claim. So yeah, we're in the final step, which is exciting. And so in addition to that, what I'm overseeing, what I'm working on is a very unique assay that can hopefully more sensitively diagnose the patients and also monitor therapy better than what's currently available. Because what's currently available is just not good.
So I understand you're not supposed to talk about molecular details of the assay, but is it like something you inject into the person? And like, what is its job? What are you assaying? What are you trying to test for? And how does it test?
So what we're doing is I can't disclose the collaborator yet, but one of the major, largest clinical institutions in the country is who we're partnering with to take our blueprint of what we use in the clinical trials and try to scale it down so it's more affordable, it's more accessible, and has higher throughput, meaning that it can be used with more patients more frequently and just more cheaper. And so we're taking that blueprint, we're trying to demonstrate that the methodology that this hospital is going to implement is equivalent or has less or similar covariance to what we use in our clinical trials, so it can appease the world and you can know that it's sensitive and it does what it says it does. It reports what it claims it reports. But to your question, it's not an injection, it's a very simple blood draw. You just take a couple of milliliters, you look at the blood and you can say, yes, this patient has Wilson's disease. And then after that, when they start treatment, you can still take blood and say, according to your levels, you're on the right path, or we need to do some sort of dose titration.
So Wilson's disease is genetic, right? It's not something that you can cure, but you treat it. And so you can monitor how well they're responding to treatment that way?
Hopefully, we're working on it. Yeah, we're trying to get there. Yeah.
Okay.
But fun fact, you actually, if you were somewhere in like Asia, such as India, you can get pseudo Wilson disease because they have so much copper in their utensils and their cooking ware, and they can get copper toxicity.
Gotcha. So just having too much copper, you can have Wilson's disease symptoms. Has this ever shown up?
Yeah, but it is genetic. It's genetic. Yeah.
So is the assay just testing for copper in the blood, or is it testing for something else?
It's definitely testing for copper, but in proportion to some other things. Like when you go to the doctor and you want to see your kidney function, sometimes you'll get creatinine clearance rate. You'll get these ratios that show a certain analyte in proportion to another, because there's one thing in the body that's constant, and it deals with your renal or your some sort of metabolic clearance, and then your other analyte could be affected in a certain scenario. So you use ratios in proportion to show the function of the organs in your body.
Gotcha. It's all relative. So what's your role in these clinical studies? What do you do in a day at work?
It changes sometimes. So it depends on the stage of the study. So in the beginning, I'll be on the team that creates the design of the trial and tries to put on paper how we want to articulate or prove our hypothesis.
So let's talk about that study design. Do you do like a double-blind, placebo-controlled experiment, or what sort of study designs do you use?
So double-blind, placebo-controlled, like that's definitely up there as some of the most robust and rigorous ways to test a hypothesis. But when you have rare diseases, not common diseases like cholesterol, diabetes, or whatever, you don't have the luxury of being able to execute the trials in that manner because your sample size is going to be like 30 people.
Yeah, you don't have enough people.
It's not going to be, So sometimes you have to have it like either investigator or sponsor, unblinded, but the patients can be blinded. You have to kind of mix and match. Yeah. But to your point, we always try to shoot for the stars and get that most robust as possible design.
What are some other study designs that you would utilize?
It's so another important concept to understand is that I'm in clinical development. I'm in the phases you were talking about earlier, 1, 2, and 3, before you hit approval by the regulators. Once you get approval by the regulators, there's another department which I did my postdoc in called medical affairs.
Yeah.
And medical affairs is basically saying, we got this great product out there. Let's answer the questions we don't know yet.
Right.
And so when you get into those types of studies, I'm not going to say they're not as well designed. They're just answering different questions. So you don't have to be as robust with your design. So one of my favorite designs that, once again, it's not that it's not as robust as like a double dummy blinds, whatever. It's just answering a different question. They do, because there might be other products in the market that treat the same condition your product treats. They do what's called a crossover. The crossover design is really cool. It tries to, like, if depending on the condition and the symptoms that are being observed and the side effects that the product gives, you can define a certain time point that's appropriate to then cross over with your different therapies to try and uncover different insights or uncover different treatment effects. And what's funny is you asked, this is tying into the very beginning of this interview, a half-life. If you know a product's half-life, you can design a really nice crossover trial. Yeah.
So this actually, I'm going to bring up the Abbvie stuff that I've seen on the news recently. Have you heard about any of the news with the House of Representatives has subpoenaed Abbvie, the makers of Humira, which I've learned.
I did not know that.
So I've been doing a little research on this. Humira is the highest grossing drug in the world. So this is the drug that makes the most money out of any drug in the world.
It's the new Lipitor, baby.
Yeah, Lipitor is for cholesterol, right?
But back in the day when I believe it was Pfizer adopted the current drug pricing model, like in the early 2000s, like to the moon.
Shut up.
I'm not saying it's right.
Yeah.
And fortunately, I'm in rare diseases, so I can make these jokes because I don't deal with these kind of things. But just there's so many things that the government can't intervene, and the price just went soaring.
So this is what
some price gouging.
This is what the government's getting involved in now, because in Europe, a dose of Humira, which, well, first, for anybody who doesn't know, Humira, you've probably seen commercials on TV. It comes in like a pen, and you inject yourself in the leg. For most people, it's like twice a month, and it treats a bunch of inflammatory conditions. So rheumatoid arthritis, I think, was the thing it was originally designed to treat. But now it's been approved for Crohn's disease, ulcerative colitis, and some other like autoimmune inflammatory conditions, right? It's a TNF alpha blocker, so it blocks a molecule called tumor necrosis factor alpha, which we know contributes to inflammation, right? So in Europe, a dose of Humira right now costs like around $500, depending on the country. In the United States, it costs about $2,500, so like 5 times that. And this has to do with patent laws. So I found out that in Europe, there's like a limit to how many patents you can submit for a medication. In the United States, there's no limit. So Humira's original, came out in 2003. The original patent expired in 2016, but the makers, Abbvie, have been spamming the patent office since then. They submitted 247 additional patents for Humira since then. And according to this lawyer, it's like a war of attrition, where you just like spam the patent office so much that like no other manufacturers can create a similar drug. So, and you can correct me if I misspeak on this, but for a drug like Humira, you can patent the process to make it, right? Because it's made using monoclonal antibodies, as opposed to like a simpler drug that's like, say Tylenol. You can't patent Tylenol, right? Acetaminophen. So you can have generic versions. Whereas a drug like Humira, you can patent the process to make it because it's so complex. And so then companies can make like biosimilars where it's a similar process. But for some reason in the United States, like Abbvie has prevented a lot of other companies from doing that. So literally Humira is like the only game in town for that sort of a drug, right?
Oh yeah, that's perfect. You said it To the T.
So I was on this drug for a little bit. Yeah, talk more about biosimilars.
Yeah, biosimilars are a lot trickier than small molecules. I'm working on a small molecule, which is a tablet. Okay. It's just a pill. It's just a capsule.
How big is the molecule?
The biologics.
Yeah, talk about how big is like a small molecule. How many atoms are you talking about?
Oh man, I love this. Is going back to the coffee, right? So like, if the caffeine is the small molecule, the like ice is the biologic.
Right? Yeah.
Yeah, like the ice is just the biologic, the protein, the monoclonal antibody. What you're referring to, it's a monster.
So insulin, like thousands of atoms long, right? Or thousands of atoms.
Yeah.
Whereas like a small
molecule
probably
doesn't even. Yeah.
So where would the cutoff be? Like maybe 100 atoms when you're going from a small to a large?
Well, the thing is, I think insulin's a great example because, I mean, let me just say that the current status of pricing of insulin is still messed up. But if we go back, so I'm talking the 40s, the 50s, when insulin first started coming out, and when we started getting better with insulin, this is and it was actually affordable. This is a great example that is easier to understand than like just pulling out random TNF alpha inhibitors, like you were saying, like the Humira adalimumab. So like if you think about all the amino acids that make up insulin, and you just take the last one on like the, I don't know, N terminal or whatever terminal, truncate it, and you find that it's absorbed better, has better efficacy, this, and this, X, Y, and Z, you can claim it as a different molecule. You can claim it as a different entity because you
cut off
change
one
the chemical structure.
Yeah.
I believe if I go back to like my notes from school, but like there are some forms of insulin where literally you're taking 2 amino acids, 3 amino acids, just truncating it, making it different, adding a salt to it, helping it with its pharmacokinetic profile, so absorption, distribution, metabolism, excretion. That makes all the difference. And not only that, because remember we're talking about biologics here, not a pill or a small, chemical. Not only the difference of a couple amino acids, but the process in which you get there.
Right.
Think about the chemistry, the chemistry scientists. They're mixing the big vats.
Right.
They got all the solutions. They have the denaturing this, the supernatants, the this and that. The whole process of just developing it. That the temperature, like everything plays a role into how the product's going to come out to be. So I'm not justifying all these spamming patents that you see, but there is some validity to your intellectual property rights when you create a more efficient and better method.
But on the other hand,
like, let's go back.
Yeah, protein. On the other hand, proteins are made naturally by the body, a lot of them, right? So if you're cutting, a new form of insulin, it's very similar to something the body naturally produces. So it's kind of a moral gray area, I suppose.
It's interesting because, we do make a lot of proteins, but a lot of what's applied therapeutically is altered for benefit. Yeah. It's altered because it's more efficient. It's better. If I gave you bovine, beef insulin, porcine, pork insulin, and then human insulin without modifying the human insulin, you're going to have better results with the animals. You're going to have better, so just because it's made by the body, if it's altered and made better, it's a different beast. It's a different thing.
Cool. I was gonna say, so one other interesting thing from like the video I saw of the House, where the House of Representatives grilling the executives from Abbvie, they had like a breakdown of their expenses, and you know how politicians a lot of times they're rhetoricians. They want to make a persuasive argument. They don't consider both sides of it. And so one thing she mentioned is that the company's major expense of like $50 billion in 5 years was dividends to shareholders and stock buybacks, which is like an economic thing where like when you're that sounds pretty egregious, but when you're a publicly traded company, you owe your investors dividends. That's part of the deal you make with your shareholders, right? And then stock buybacks, I figured out, is when essentially companies buy some of their stocks off the market to keep prices high. So that's like arguably, it has nothing to do with medicine. It's just the company trying to like keep their stocks high to please their investors. Could be a necessary evil though, right? Because like these companies with all their investors, they do come up with a lot of good drugs. But the thing that I think that the politician was kind of misleading on is Abbvie spent like $2.8 billion on research and development for Humira between 2013 and 2018. And she kept saying the drug didn't get any better between that time. But you know what actually changed with Humira between 2013 and 2018?
The salt form? The solution that
I don't know if they changed the actual drug, but they got it approved to treat a lot more conditions. So like my condition, ulcerative colitis, was not what Humira was originally designed for, right? But obviously, to get it approved to treat ulcerative colitis or any new condition, you have to run
takes so much money.
Rigorous clinical studies like you're doing. So I think that was really either ignorant or dishonest on the politician's part to say like, this is wasted money at R&D, because in order to treat a new condition, you obviously have to do a lot of sound research on the drug, and that costs money. So
definitely, all the unethical noise aside, like all that unethical trash of the pharmaceutical industry, which it happens. All that aside, like
it's a business.
It is, but there are so many benefits from the amount of money that's being poured in, and people are living better and longer lives.
Yeah.
And like I said, I acknowledge all the trash and all the unethical issues.
Hopefully we can.
At the end of the day, at least with my work, I like to focus on the more altruistic nature, like people that were dying previously, like in rare diseases that had no hope, now have a longer, better life.
Right, or somebody with Crohn's or ulcerative colitis, these medicines can be a real lifesaver, literally. It's not. We don't need to. Yeah.
It's like you could hold me to it in this interview. Like since the start of my internship at New Haven in Alexion, like then my postdoc at Alexion, like I want to stay in rare diseases. I really don't want to move away from drug development in rare diseases. It just, to me, gives me the peace of mind and the altruistic nature to continue working on what I'm doing.
Well, Dr. Quicquaro, I'm going to have to end today's interview here. Thank you so much for your time, and maybe sometime soon I can pick your brain more about like vaccines and drug trials and some other things we didn't have time to talk about. But thank you so much, and I'll be posting this up soon. All right, take care, buddy. Love you.